Okay, team, let's finish up this chapter, Concept 14.4. Let's take it all back to human beings because, by golly, it's nice to talk about us because we understand us and we tend to think that we're the most important species on the planet, so why not bring all this information back to us? Mendel's garden peas were very cool and he learned a lot of cool information, but perhaps it didn't apply perfectly to humans. And what can we know, or what do we know now about human genetics and some various human traits and diseases that we can talk about that kind of relate back to perhaps what Mendel learned? So concept 14.4 says many human traits follow Mendelian patterns of inheritance. So we are not peas, but we said that a lot of the information that Gregor Mendel learned is applicable to any living creature that has a sexual reproductive lifestyle, and that would certainly be humans. So some of those same ideas and traits apply to us. Now the problem is that humans are not very good scientific subjects, particularly for genetic research, because the generation time is too long. You're talking probably 18 or 20 years between generations. Who wants to run an experiment for 20 years just to get a single generation? Parents produce relatively few offspring, one or two at a time, which means you have to have enormous numbers of parents to produce large quantities of data. And then breeding experiments are unacceptable. For some reason, people frown on this idea of breeding humans to other humans just to do their scientific experiments and find out what might happen. So that's usually looked down upon, so we try to avoid that. So even though human studies are not widely done the way we can do studies on pea plants or animals, we do know that some of the basic Mendelian genetics applies to us and endures as the basic foundation for what we understand in human genetics as well. So what can we do? Because we can't pull, hundreds of humans into a lab and have them breed and test their babies. So we oftentimes have to kind of work in reverse. Instead of doing breeding programs and studying the outcome, we have to look at breeding pairings and matings that have already occurred. And so we're going to look backwards, of course, and we're going to do what's called a pedigree. A pedigree is basically a family tree that describes all the various interrelationships between parents and children across multiple generations. And you see this a lot in, could be fancy horses or fancy dogs, or nowadays they even have internet sites for people who like to do work on their families, and they go back and they have websites where you can go and put in your information to find out who was your great 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 grandfather, or were you related to someone famous or someone notorious in history, so you can learn more about your ancestry. That used to be pretty hard. Genealogy used to be difficult. You used to have to go to the courthouse and look up dusty documents and try to track down and talk to people in different parts of the country. But now websites make genealogy research quite a bit more easy and perhaps a bit more fun for just the average person. So you would find a pedigree, basically your family tree, and that can show inheritance patterns. So you can look at the people who have come together and had children, and you can look at their various traits, and you can see the patterns that arise via genetics. So we can still learn a lot of information by looking backwards. So let's start with these two ladies right here. They are sisters. They are the third generation that we're going to look at. And notice that one of them has what's called a widow's peak. Right there, her hair comes to a point at the forehead, and hers does not. Hers is kind of straight across. So a widow's peak or a non-widow's peak. And how would we determine, if we didn't know ahead of time, if the widow's peak was dominant or recessive? Well, we can actually look back in her family and determine quite a bit of information. So we know that, or we're going to find out that the widow's peak, no widow's peak, is recessive, and the widow's peak is going to end up being dominant. But how might we figure that out? Well, we could go back in time. So let's look at the grandparents of these two daughters. We go back and look at the grandparents. This particular male is affected. In other words, he is with the widow's peak, and grandma here is without. And then this male, grandpa, is without, and this female, grandma, has the widow's peak. Now, if we follow the children that they had, and the children that these grandparents had, these two children eventually met and married, and they became the parents of these two daughters. We can determine by looking back which trait is dominant, which trait is recessive, and we can see the variations and the relationships between the parents, the grandparents, the parents, and then the children. We can do the same by looking at another trait. So attached earlobes here and free earlobes. And again, we can go back in time, looking at the parents and the grandparents, and we can start to make some determinations based on the pedigree that we see. So once we know the pedigree, not only can we work backwards to determine certain things like dominance and recessive, then once we have that information, we can make future predictions about the future children. So we can make predictions about these two young ladies, if we know the genetics of the person they might marry, what their children might be. So we can kind of go forward and backwards using that pedigree. And then we can use our various probability rules of multiplication and addition to predict the probability of specific phenotypes. So here's a good question. What would be the chance that a third sibling of the two young ladies we looked at would have both a widow's peak and attached earlobes? Well, we can do that fairly easily. We know by going through the genetics, by going through the Punnett square, or the probabilities, that these two traits, first off, they have nothing to do with each other. They're not connected. So their probabilities are going to be figured separately, and then we can add those together. Or actually, we're going to multiply them together because we want to know what are the chances that both of these traits will show up at the same time. So if we take a look and go back to our chart, so we have the widow's peak, which is going to be the W, and the attached earlobes, which are going to be the F. We have to go back and look at their parents here. So both mom and dad for widow's peak are heterozygous, big W, little w. And for the earlobes, they're also both heterozygous, big F, little f. So we can take a look at the chances of that. And with this particular cross, the widow's peak has a 3/4 chance, because it can either be the homozygous dominant or the heterozygous, which is going to be 3 out of the 4. So having the widow's peak is a 3/4 chance. Having the attached earlobes is only a 1/4 chance, because you have to be homozygous recessive to have that. So we can look at these two separately. The widow's peak is a 3/4 chance. The attached earlobes is a 1/4 chance. When we multiply those two together, we find out the possibility of having both of those at the same time. And so it's a 3 out of 16, a 3/16 possibility that the next child of those parents, which would be either the sister or brother of the two girls we looked at, would have both a widow's peak and attached earlobes. So we don't have to do massive lab experiments using humans. We can do some of this work using a pedigree, looking back on matings that have already occurred and getting our information that way. Sometimes there are genetic occurrences that occur in humans that are not innocuous or harmless, like whether or not you have a widow's peak. That doesn't really affect your lifespan or affect your health or anything like that. But there are some genetic issues that do quite a bit affect your health, could cause your death, or could cause a shortened lifespan. A lot of these disorders are recessively inherited. Many genetic disorders, particularly in humans, are inherited in a recessive manner, and you would only have that problem or that disorder if you're homozygous recessive. A good example is albinism. If you are an albino, you lack pigmentation in your skin, in your hair, in your eyes. That's a recessive trait. You only have that condition if you receive that allele for that trait from both mom and dad, the recessive allele from both mom and dad. Because having coloration, having pigmentation is the dominant trait. So you can have that if you are homozygous dominant or heterozygous. That's what's going to show up most often. Some of these recessively inherited disorders are fairly mild, and others can be quite damaging, even life-threatening. Like we mentioned, recessively inherited disorders only show up if you're homozygous recessive. That makes them a bit more rare. And if you are heterozygous for one of these disorders, we call you a carrier. So you have the possibility of passing on that genetic recessive allele to your offspring, but you would never exhibit that trait. So you might not even know that you were a carrier for that issue until you had a child that turned out to have that issue. And that might be the case if both you and your husband could be carriers. If you were both carriers, there's the potential that in your child, the homozygous recessive will meet, and then that particular genotype would show as the recessive phenotype. And as I mentioned, albinism, being albino, lacking pigmentation, is an example of that. The only way that that occurs is if both parents happen to be carriers, they're heterozygous, or perhaps both parents are albino themselves, homozygous recessive, or perhaps if one parent is a carrier and one parent is albino. Again, all of those create the possibility of having the small a homozygous recessive alleles. In humans in general, these genetic disorders are not evenly distributed among all humans across the world. Certain disorders tend to be concentrated in certain populations due to genetic histories when these various populations were more separated. So you may have certain disorders that are more prominent in African Americans, or you may have certain disorders that are more prominent in Europeans or Native Americans, because those populations used to be further separated from each other, so they evolved different genetics, and so they also evolved different mutations or different disorders. So for example, you might have sickle cell anemia, which tends to be more prevalent in African Americans, or you might have Tay-Sachs disease or albinism, which tends to be more prominent in certain groups around the world. If a recessive allele that causes the disease is pretty rare, well, then there's a very slim chance that two carriers who don't know they have that gene are going to meet, marry, and have that child. That would kind of be shocking, but it does occur, of course. It can be more likely in matings that are between fairly close relatives. We call these consanguineous. Consanguineous matings would be matings between fairly close relatives. This is basically creating a smaller gene pool. So if there is a particular mutation or a particular disorder, even if it's a recessive disorder, it's more likely to show up because you are basically concentrating the genetics, both good and bad. Most societies and cultures around the world have laws or social taboos against marriage between close relatives. And some of that may have resulted from just their basic observation that that was problematic, that when close relatives married and reproduced, that oftentimes, more often, the children had various physical deformities and difficulties. So some of that's going to be related to perhaps scientific observation. Some of that may be related to social factors, economic factors, religious factors. That all plays a role in that as well. A good example of this kind of shows up when you talk about royal families and royal bloodlines, especially not so much now, but in previous history, a lot of times the various royal families intermarried quite a bit, and so you had those who were fairly closely related to each other, or certainly more closely related than the common person, marrying each other, and they tended to have oftentimes various traits, some of which were quite detrimental, that ran in the family because they had such close pairings between bloodlines. In modern times and here in the United States, of course, those close relationships are fairly rare. So you have a greater mixing of the population, and now we have world travel, so you have people from all over the world that meet and fall in love and have children with various people. So you have a much better mix of genetics. It makes it more rare that you're going to get an example of one of these homozygous recessive traits pop up. So let's talk about a couple of them. One of them that is unfortunately somewhat common is called cystic fibrosis. Cystic fibrosis is the most common lethal genetic disease in the United States. It strikes about 1 out of every 2,500 people, so that would be about 4% of those of European descent. So it's slightly more prevalent in people of European descent than it is in other populations. And again, this goes back to what we were talking about, how at one time various populations were more isolated, so they developed characteristics that might differ from other populations. In cystic fibrosis, the allele that is defective results in the absence of a chloride transport channel in the plasma membrane. So think back when we were talking about the cells. Remember, there are transport channels that allow certain ions or chemicals to pass through the plasma membrane. Well, cystic fibrosis creates a defect where the transport channel is not there or it doesn't work very well. And what this means is you have a buildup of chloride ions outside of the cell. The effect of this problem is you have symptoms that include a lot of fluid buildup on the lungs, mucus buildup in some of the internal organs, which leads to you not being able to digest and absorb nutrients very well. These people who suffer from cystic fibrosis tend to have chronic bronchitis, recurrent bacterial infections. They oftentimes have to have various treatments to try to drain fluid out of their airways and help them live longer and healthier lives. If this disease is completely untreated, most of the life expectancy tends to be very short, 5 or 6 years. But if it's aggressively treated with antibiotics and a variety of other therapies, a lot of people with cystic fibrosis can live a fairly long life, can prolong their life into their 20s and 30s and have a good functioning life. But they do have to constantly work to keep the symptoms of this particular disorder at bay. Another good example of a disease, genetic-related disease in humans, is sickle cell anemia. Sickle cell disease is a genetic disorder that is interesting because it has a particular evolutionary implication that we'll take a look at. It's more common in African Americans, again going back to this idea that these populations tended to be separated back further in time, so they developed both good and bad mutations in their genetic pools. So sickle cell disease affects 1 out of every 400 African Americans. So again, not common, but not completely uncommon either. And this disease is caused by a substitution of a single amino acid in the hemoglobin protein in a red blood cell. If you remember when we talked about proteins way back in, I don't know, it was like chapter 2 or 3, hemoglobin is a very complex, twisted and folded protein that carries oxygen in the red blood cells. Well, you have a single amino acid chain, 1 amino acid out of the entire chain that makes up this protein, and that causes a problem with the red blood cells. If you are homozygous for this trait, you have the disease and or the condition, and your red blood cells oftentimes are sickle cell shaped because there's a misfolding of some of the protein structure, and it causes a change in the hemoglobin. So right here you see the sickle cell shape of someone who is affected. If you are heterozygous for the trait, you will have a combination of both normal blood cells and sickle cells. Symptoms include physical weakness, pain, organ damage, even paralysis, and can lead to death if it's untreated. These sickle cells cannot do a very good job of carrying oxygen. They tend to get clogged in capillaries and cause a variety of problems. As I mentioned, if you're heterozygote for this allele, for this trait, you don't have the sickle cell disease, but you have what's called sickle cell trait. That means you kind of partially suffer. You may suffer some of the same symptoms, but not to the degree that someone who has the full homozygous recessive alleles may have. If we look at African Americans, about 1 out of 10 African Americans has sickle cell trait. In other words, about 1 out of 10 African American people have, or at least are heterozygotes for this particular trait, and that's extremely high. That's a very high frequency of an allele that is very detrimental when it's a homozygote. So the question is, why would this be? Because when this allele is in the homozygous recessive, it can be deadly, can be very detrimental. So why would there be so many carriers? Why would evolution not weed that out and make it less common? Say, for example, like cystic fibrosis or something that tends to be far less common. You're not going to find 1 out of 10 being heterozygous for cystic fibrosis. So why hasn't evolution weeded out this allele? That's a very good question. And it turns out the reason has to do with evolution. Think about the population where sickle cell tends to be more common in African Americans, or in populations that originated in Africa or around the equator in the tropics of Africa. In those areas, there's a disease called malaria. It's caused by an infection. It's a disease caused by a little parasitic protozoan, and it's carried by mosquitoes. So a mosquito will bite a human or another creature that's infected with this protozoan, will suck up some of that protozoan in the blood that it gets, and then it will fly off and bite someone else. And when it injects its little mouthpiece into you, some of that other person's blood and some of the parasite from that other person or from that other creature will actually go into you, kind of like backwash. So now you're infected. So in tropical areas in Africa, malaria is a very widespread infectious disease. It's one of those diseases and problems that we have been trying to tamp down and eradicate for many, many years. As a matter of fact, you may have heard of like the Bill Gates Foundation. That's one of their primary goals in the area, is to distribute mosquito netting and medication to try to combat malarial infections, which are fairly rampant. Well, here's how that deals with sickle cell anemia. It turns out that if you are a heterozygote for sickle cell, meaning you have some sickle cells and some normal cells, you actually are less susceptible to the parasite. So it turns out that there's actually a benefit to having this gene. Now, you don't want to be homozygous recessive or you have full-blown sickle cell, and that's not good. There's too many detrimental causes or too many detrimental things there. But if you're a heterozygote and you live in Africa where malarial infections are very prevalent, you actually have an advantage because the infections for you tend to be less common and they tend to be less severe. So evolution has kept this around because in one way, it provides a benefit, especially if you are a carrier and you don't have the full-blown disease. So that's very interesting. So how genetics and environment play a part in what's going on here. Let's kind of finish up by looking at a couple of other examples of human genetic issues. There are some inherited disorders that are dominant. So they are dominantly inherited disorders. That's a little bit more rare, but it can happen. So some human disorders are caused by dominant alleles, and they generally arise by mutation, as do all lethal alleles. They arise by mutation. That's the genesis of these various different disorders and problems. And the reason that a dominant allele that causes a disorder doesn't tend to stick around as long is because if the lethal allele is dominant, it's going to show up every time, and that means that particular person is probably not going to be able to live long enough to reproduce. So a lethal allele has a 50-50 chance of being dominant or recessive, but recessive alleles can be hidden. They can be passed on via carriers who may not have the full-blown disorder, where dominant alleles, if they're there, they're going to show. And if it's a fairly significant disorder, it may cause the death of that individual long before they can reproduce and pass it on. So dominant disorders tend to be fairly rare. They have to be disorders that are not particularly lethal, or they wouldn't be around for very long. A good example of a dominantly inherited disorder is achondroplasia. I'm not sure if I'm pronouncing that correctly, but it looks like achondroplasia, and that's a form of dwarfism caused by a fairly rare dominant allele. And so, for example, you have a doctor here, in the textbook it even talks about this particular surgeon or doctor, and of course he has this condition, and he studies this condition in other people to help those people. So this is not necessarily a lethal condition, which is why it continues to be around in the population as a dominantly passed on genetic disorder. Another example of a dominant allele disorder is called Huntington's disease. And again, this is more rare, but the reason that Huntington's disease has stayed around and hasn't been kind of weeded out via evolution is because the symptoms don't show up usually until later in life. So the timing of the onset of the disease has a significant effect on its inheritance. Huntington's disease is a degenerative disease of the nervous system, and oftentimes you don't even see any effects until you are middle age and older, 30, 35, 40. And by the time you see the effects, by the time you discover that you have this disorder, you've likely already had children. So you either have or have not already passed on that allele before you ever knew that you had the problem. And once this deterioration begins, it tends to move fairly rapidly, and the condition is irreversible and ends up being fatal. So sadly, this is almost kind of like a genetic time bomb within yourself. You probably get married and have children, and perhaps you're in your mid-20s or early 30s, and you've already created 2 or 3 children. And then at 35 or 40, you develop this condition, this disease, and it turns out you had it all along, but the symptoms didn't manifest themselves. And now you've already produced your children, and the question is, did you or did you not pass on those genetics to your children? Modern genetic study of this disease has actually located the location, the locus of this particular allele, and sequenced this gene in about 1993. What this means is we now have tests to detect the Huntington's disease allele before symptoms develop, before you decide whether or not to have children. So that gets to kind of a question of, would you want to know? I mean, if you are worried about this particular condition, do you want to be tested, say at age 23 when you first get married, to find out? Or do you just want to take the chance and have your children and then perhaps find out later? Now, this is a genetic disease, so it would tend to run in families. So not everyone in the world population should probably run out and get this test done, because it's very, very unlikely that you would have it. But if Huntington's disease was at all present in your family, that might be something to consider before you had children, whether or not you want to know if there's a possibility of passing this on. Finally, as far as genetic diseases, let's talk about what's called multifactorial disorders. Remember, multifactorial means there's a lot of things that affect it, genetics and environment. Lots of human diseases, such as heart disease and diabetes, and alcoholism, mental illness, cancer, they have genetic and environmental components. So the problem is, it's very difficult to determine what is the cause of this problem, because there are so many different things that play a part. Genetics may play a big part in whether you are at a higher risk for mental illness or a higher risk for alcoholism. But obviously your environment plays a part. You could have a great genetic propensity toward alcoholism, but if you never take a drop of alcohol in your life, well, that won't be a problem for you, and you avoid that issue. You may have in your family very high rates of heart disease, but depending on your diet and exercise habits and whether or not you smoke, and you can either lower that risk or increase, make it worse based on your environment and other factors. So there's not a lot that's understood about the genetics of multifactorial diseases, because there's so many things that affect it. So it's probably, for the time being, until we learn more about the genetic component, it's probably a good idea just to encourage people to make proper environmental choices, such as eating healthy and exercising. And that way, even if you have a genetic risk, you're lowering that risk as best you can. I think Gregor Mendel would freak out if he knew what we could do today concerning genetics. There is all sorts of genetic testing and counseling that can go on. This particularly is important to parents. When you are about to have your first child or about to decide to get pregnant, whether or not you want to go through genetic testing for you and your partner, or then later have genetic testing done on the fetus that is developing inside the mother. These are sticky questions. Some people want as much information as possible, and some people would prefer perhaps not to know. Nowadays, because we know so much about genetics, we can use these rules of probability and your pedigree and your background to kind of determine what are the odds that your child may have a genetic disorder. And that may help you determine whether or not you want to get this testing done. If you are from a family that has a high percentage of a certain genetic disorder, then it's more likely that you're going to want to get tested. And then you know what are the probabilities that when you and your spouse have a child, that child may receive that genetic disorder from you and your partner. For most of human history, if you were a carrier of one of these diseases, you wouldn't know it until you had a child. And you could have multiple children who did not have the disease, because remember, you are a carrier, you're heterozygous. You may not give the recessive allele, and if your partner is homozygous dominant, you would never see it. If they're heterozygous, you still may not see it. It's just chance and probability. But nowadays we have various tests that can identify you even if you're a carrier. So you don't have to have the actual condition or disease. We can do testing that might identify whether you're a carrier, and then you can determine what are the probabilities and do you want to proceed with having children, or what do you want to do to prepare for that possibility. We can even do testing after the pregnancy has begun. So nowadays we do fetal testing. There's a couple different types of fetal testing that can happen. One of them is called amniocentesis. Amniocentesis is where they take a sample of the liquid that surrounds the fetus. The fetus is growing inside an amniotic sac, and that sac is filled with liquid that cushions the fetus and provides nutrients to the fetus as well in some degree. And so that liquid contains some of the genetics of the child as well. So a portion of that liquid can be drawn out using a needle and syringe and then be tested for various conditions. The other type of fetal testing that can be done is called chorionic villus sampling. Chorionic villus sampling, CVS, is actually sampling a portion of the placenta. The placenta, again, is part of this amniotic sac that covers, basically surrounds the baby in this bubble, and it's the placenta that connects the mother and child. So you can take a small snip proportion of that and test it for different genetic abnormalities or conditions. And then we have other techniques that are not as invasive. We have ultrasound, which you've probably seen in movies and TV before, where they take a wand and they rub a jelly-like material on the woman's belly, and then they use this wand to produce an ultrasound picture, kind of like a radar screen, that shows the fetus and can show physical problems or various issues that you might be able to see inside. And then there's I can't say this word, fetoscopy. Fetoscopy is probably how you pronounce it. That is actually a way of using a particular device where you can look inside at the fetus itself and see various things that might be helpful in determining whether there is a problem. All these tests are good in a way in that they provide helpful information, but then of course the downside is whether or not you want to know, and how much information you want to know, and how is that going to affect your decision of what to do. Because of course if you find out that your fetus that would become eventually your child has a strongly lethal genetic disorder, that may mean that you miscarry, or that the child dies very shortly after birth, or has enormous physical difficulties. You may decide that you want to have an abortion, or you may want to decide that you don't have the ability to care for that child, or you may decide that there are preparations you need to make to care for that child, or you may decide that that's fine with you and you want to go ahead with that pregnancy. So this information is good, I guess, but it also can result in a lot of difficult choices for parents. Perhaps choices that people 100 years ago didn't have to face. So it's a double-edged sword. They didn't have the information and couldn't prepare, but they also didn't have to go through some of the difficult decisions that modern parents-to-be have to do. This clip is a good example of an ultrasound of a human fetus. Probably something you've seen before if you have any relatives or friends of your parents that have ever had children. They've probably shown you pictures taken from the ultrasound. So if you want to click on this, you can play it if you have the PowerPoint. I think I might have thrown this video clip up in the notes as well, and if not, I can put it up there. So this would just be an ultrasound. This is not an invasive process at all. This is where they rub the jelly material on the woman's belly, and that's just to get good contact between the ultrasound wand and her skin, and then they basically use sound waves to bounce off the fetus inside and to get somewhat of a picture of what's going on inside there. These two graphics are kind of showing the two different types of tests I talked about. Amniocentesis, where they actually take a portion of the fluid that's surrounding the baby, and they can do genetic testing on that. That can take a certain amount of time, because they have to allow those fetal cells to incubate and grow, and then they can take a look at the genetics and create a karyotype from that. Or they can do the CVS, a chorionic villus sampling. That only takes a few hours. It's quite a bit faster, and they can do a karyotype and look at the chromosomes of the fetus. Last but not least, they can even do some genetic testing after the baby is born. So there are some what they call newborn screenings that are fairly common nowadays. One of these is called a PKU test. It tests for a particular genetic disorder in which the body cannot properly break down and metabolize an amino acid called phenylalanine. So this is not a common problem, but it's not uncommon. So it's usually given to pretty much every infant, this quick little test where they check to see if the PKU disorder is present. And this disorder is one that can be controlled with proper diet, things like that. So you may have heard like certain fake sweeteners like aspartame, what is it, aspartame or aspartame? I can't remember how to say it, but it's the fake sweetener that you might find in like diet sodas that contains a lot of this phenylalanine. And if you had PKU, you wouldn't want to drink a lot of those sodas because your body doesn't have the ability to process that material. So this is something that's commonly done for all infants. And if you have that issue or if your baby has that issue, it's something that can be controlled throughout their life. So that's it. Lots of information. It's a long chapter with a lot of information, but cool information. And I don't think it's too difficult information, just a lot of reading. So we're going to end up with this cool little picture from one of my favorite artists that works online. She does all sorts of cool prints about scientists. This is her print about Gregor Mendel, and of course his pea plant there, father of genetics. Also, I'm going to throw in and leave up these last two things here. This talks about a lot of the different information that we've discussed in this chapter, complete dominance and incomplete dominance, and multiple alleles, and pleiotropy, and all sorts of other things. So I'll leave these in here as well, and you can take a look at those. Again, look over the chapter, look at the summaries, go watch a lot of the videos that I'll link to. I think it'll be very helpful. And check us out and catch us on the online discussion as we get ready for this big exam number 3. We're going to move on to chapter 15, and I will see you back here in just a little while. Bye-bye.